Hello and welcome to Nikolai's Genetics Lessons, and today's video is going to be about bioinformatics. If you just start learning bioinformatics and such pictures like this one is a headache for you to understand, in this video I will try to explain you how to decipher information what you see here. Now let's read the problem. Shown below is a double-stranded bacterial E. coli DNA sequence coding for a hepatocytical protein. The nucleotides are numbered from 1 to 100. Note for this problem, transcription begins with and includes the red and underlined CG, top strand, bottom strand, so here. Base pair and RNA polymerase proceeds from left to right along the DNA. And here are the three questions. Which strand is used as template strand, the top or the bottom? One more time, for this problem, transcription begins with and includes the red and underlined CG, top strand, bottom strand. Base pair and RNA polymerase proceeds from left to right along the DNA. So let's add this line that would show us the direction in which RNA polymerase would proceed. Now the main question is whether this strand of the DNA is going to be template strand or this strand is going to be template strand of the DNA from which this messenger RNA would be built and would be complementary to. Take a look. If we'll consider that this top strand of the DNA would be template strand of the DNA, we have 5' end here, then here we are going to have 3' end and 5' end of the messenger RNA here. So it's going to be complementary to this top strand. So our messenger RNA would be built from 3' end to 5' end. And if we consider that this bottom strand of the DNA would be template strand of the DNA, we have 3' end here, and then we should have 5' end here and 3' end here, if this messenger RNA is going to be complementary to the bottom strand. And actually, all you have to know in order to answer this question, that messenger RNA always built from 5' end to 3' end direction. So we can say that bottom strand have to be a template strand of the DNA. And top strand of the DNA we call coding strand of the DNA. And the second question, where would the promoter be relative to the start of the transcription? So here is the start of the transcription. If our messenger RNA we have found growing to the right side from 5' end to the 3' end, then promoter sequence is going to be to the left where transcription begins. Now we have answered first two questions, so the answer to the first question, bottom strand of the DNA, is template strand of the DNA, and the answer to this question is going to be to the left, so left. And the last question, what are the first 15 nucleotides of the resulting messenger RNA? Indicate the 5' end and 3' ends of the messenger RNA. One more time, this is going to be our first nucleotide. Here is going to be 5' end of the messenger RNA, and here is going to be 3' end of the messenger RNA. Now the sequence of the messenger RNA. Because this is going to be template strand of the DNA, our messenger RNA is going to be complementary to this sequence. In double-stranded DNA, adenine base pair with thymine. In messenger RNA, thymine would be substituted with uracil, and guanine base pair with cytosine. Now let's write down the sequence. So this guanine would base pair with cytosine. So cytosine. Let me increase the picture. And as you see, adenine would base pair with uracil. In double-stranded DNA would base pair with thymine, but in messenger RNA with uracil. So we put uracil here, and thymine would base pair with adenine. We put adenine here, adenine, uracil, adenine, uracil, uracil, guanine, uracil, guanine, adenine, and guanine, and adenine, uracil. Now let's count how many bases do we have. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15 bases. 5' end is going to be here, and 3' end is going to be here. You already know that bottom strand is template strand of the DNA, and I said that top strand is a coding strand of the DNA. Now you can say why. Take a look. If we have cytosine here, we also have cytosine here. Next, thymine is substituted with uracil in messenger RNA, so we have uracil here, adenine here, adenine here. Thymine substitute with uracil, and adenine here, uracil, uracil, guanine, uracil, guanine, adenine, guanine, adenine, and uracil here. So you see, basically the sequence of the messenger RNA is the same as the sequence of the coding strand. Also, please pay attention that 5' end of the messenger RNA and 3' end is the same as 5' end of the coding strand of the DNA and 3' end. So messenger RNA has the same sequence as coding strand of the DNA, and also directionality is the same. Hence the name coding strand of the DNA, and this is template strand of the DNA, because the sequence of the messenger RNA is going to be complementary to the template strand of the DNA. And couple more interesting notes. Numbers here, 20, 40, 60, 80, and 100, just help you to understand position of the nucleotides. So here is the 20 nucleotides, here is the 40, 80, and 100. And pluses here just show you also positions, but with a precision of 10 nucleotides. So this plus at the position 90, and this plus at the position 60 plus 10, 70, and this plus here at the position of the 30 nucleotides. I hope you have found this video interesting and helpful, and see you in the next video. Goodbye.